Hi, thank you for attending my presentation today. My name is Lisa Beppu, and I'm coming to you from the University of Pittsburgh School of Medicine. Today, I will be discussing the role of transcriptional regulator BLIMP1 in adipose-resident Tregs and how BLIMP1 positive Tregs control adipocyte browning and obesity. So, before I start talking about adipose Tregs, I'd like to take a step back and briefly cover obesity, which is the framework in which we will talk about these cells today. Obesity is clinically defined as having a BMI of 30 or greater and an excess of visceral fat around your abdominal organs. When we talk about obesity, our tendency is to attribute it to an energy imbalance resulting from poor diet and a sedentary lifestyle. But it's actually a multifactorial disease caused by many biological, genetic, social, and behavioral contributors. Furthermore, obesity is a major health concern because it's associated with a number of chronic comorbidities such as depression, type 2 diabetes, cardiovascular disease, and even cancer. And if we study the epidemiology of obesity, it's very evident that this is an epidemic that isn't going away anytime soon. As of last year, over two-thirds of the country had an obesity rate of 30% or higher. Given this information, it's evident that there is an urgent need to find therapeutic interventions beyond altering diet and exercise that can help alleviate some of the complications linked to this disease. When we talk about adipose tissue, the first thing that typically draws our attention are the adipocytes, which are specialized energy storage cells. However, in the past decade or so, we found that the adipose tissue is actually a reservoir for innate and adaptive immune cells, and that crosstalk between immune cells and adipocytes can help regulate metabolism. For example, in lean adipose tissue, you see that it's populated primarily with anti-inflammatory immune cells that secrete things like IL-10, IL-4, and TGF-beta 1 to help maintain insulin sensitivity and glucose tolerance. However, in obese adipose tissue, there's enrichment of inflammatory immune cells and shrinkage of anti-inflammatory populations such as Tregs, creating an inflammatory cytokine milieu that worsens insulin resistance and glucose intolerance. So much of the research in adipose immunobiology so far has focused on identifying adipose-resident immune cells and their link to insulin resistance and glucose intolerance. However, what has been less studied are the transcriptional mechanisms and machinery dictating differentiation and effector function of these cells. But this is slowly starting to change. In 2016, the Mathis lab published microarray data from Tregs of various peripheral tissues, including the visceral adipose tissue. And what really stood out to us was this gene PRDM1, which encodes for BLIMP1, which is the topic of our talk today. And PRDM1 was highly upregulated in VAT Tregs compared to Tregs from other organs. And the reason that PRDM1 caught our attention is because BLIMP1 is known to interact with ID proteins, and our lab has previously established a role for ID2 in adipose Treg maintenance and survival. So what is BLIMP1? BLIMP1 is a transcriptional regulator that can either promote or repress expression of various genes that are important in development, differentiation, and effector function across a wide range of innate and adaptive immune cells. Specifically for Tregs, BLIMP1 is known to positively regulate the expression of anti-inflammatory cytokine IL-10. So now that we have an overview of BLIMP1, adipose Tregs, and obesity, our lab wanted to ask, is there a role for BLIMP1 in adipose-resident Tregs? And our major overarching hypothesis was that BLIMP1 is required for differentiation, survival, and function of these cells. So to formally test this hypothesis, we took advantage of PRDM1 FOXP3 Cree conditional knockout mice, which lack BLIMP1 specifically in Tregs. And we placed these mice on 60% high-fat diet and analyzed them after 16 to 18 weeks. As a control, we also analyzed age-matched mice on standard chow. However, for the sake of time, I'm only going to be covering the high-fat diet data during this presentation because we're really interested in how these cells perform during obesity. However, if you'd like to see some standard fat diet data, I have some available on my poster, so please drop by. So to get started, the first question we ask is, does loss of BLIMP1 affect total adipose Treg frequency? And to find out, we isolated epididymal visceral adipose tissue and inguinal adipose tissue from BLIMP1 conditional knockout and littermate controls, and looked at the percentage of CD4 FOXP3 positive cells via flow cytometry. And we found that there was no difference in the frequency of total Tregs in both types of tissue. Next, we examined whether BLIMP1 plays a role in adipose Treg differentiation. It has been reported before that BLIMP1 can play a role in preserving effector Treg identity. However, this has been demonstrated to actually be tissue type dependent. To investigate, we used flow cytometry to measure ST2 KLRG1 positive Tregs and found that BLIMP1 deletion results in reduced frequency of Tregs positive for these proteins. Although not shown here, this was also true for a number of other effector Treg markers, and thus we concluded that BLIMP1 is required to maintain the adipose effector Treg signature. Finally, we wanted to see if BLIMP1 plays a cell-intrinsic role in regulating ALT10 production, as this has been observed previously in other types of immune cells. When we isolated the stromal vascular fraction from adipose tissue and stimulated them ex vivo, we found that in the absence of BLIMP1, fewer adipose Tregs produced ALT10. So up to this point, all these data were somewhat expected based off the existing literature. However, what we observed next was quite surprising. One thing that stood out to us was that when we placed our mice on high-fat diet and observed them over the course of several months, the conditional knockout mice gained weight at a slower rate compared to the raw-type counterparts. In addition, when we measured insulin resistance, we found it to be lower in the conditional knockout mice compared to the control mice. We couldn't really ignore these observations because it's commonly accepted in the immunometabolism field that effector Tregs are protective against insulin resistance. So how is it possible that destabilizing effector Tregs through selective BLIMP1 deletion was improving insulin sensitivity? So to help answer this question, we drew on a recent discovery by the Tontonitz lab that AL10 has the ability to suppress thermogenesis, and proposed our own hypothesis that ablation of BLIMP1 dependent AL10 signaling by Tregs induces thermogenesis to diminish weight gain and mitigate insulin resistance. To test our hypothesis, we isolated epididymal and inguinal white adipose tissue from our BLIMP1 conditional knockout mice and looked at their thermogenic profile. And what we discovered was that both fat pads were smaller in the conditional knockouts compared to the wild-type mice, and there were measurable increases in thermogenic genes and proteins such as UCP1, PRDM16, and DO2. To really be sure that thermogenesis was being suppressed through a BLIMP1 AL10 axis, we repeated all our assays in AL10 FOXP3 conditional knockout mice, which harbor dysfunctional Tregs that lack the ability to produce AL10, and saw that these mice phenocopied the BLIMP1 conditional knockout mice. For example, these AL10 conditional knockout mice weighed less, had smaller fat pads, decreased insulin resistance, and increased expression of thermogenic markers. So taken together, all these data supported the idea that during diet-induced obesity, adipose effector Tregs can signal through a BLIMP1 AL10 axis to suppress thermogenesis, and that in the absence of BLIMP1 positive Tregs, less AL10 in the environment led to increased thermogenesis, reduced weight gain, and better insulin sensitivity in mice. So this is really great, but at the end of the day, what we're really interested in is whether or not there's any clinical translational relevance. To address this point, our lab has begun mining public databases to see whether or not we can correlate BMI with adipose Tregs in humans. And what we have here is cross-sectional data from the MET-SIM study, which is a longitudinal population-based study that performs microarray analysis on subcutaneous adipose tissue from randomly selected Finnish men. And it turns out that obesity worsens with both IL-10 and IL-10 receptor expression in the adipose tissue, and more specifically, BMI goes up as you have more IL-10 expressing Tregs. So, although this is only correlative data, our preliminary findings support the notion of validating whether or not our preclinical data is replicable in humans. So, to recap our model so far, we believe that BLIMP1 positive Tregs secrete IL-10, which act on adipocytes to dampen thermogenesis and maintain metabolic homeostasis. However, we think that in the absence of BLIMP1 positive adipose Tregs, there is less local IL-10, and this leads to increased thermogenesis and slows down the progression of weight gain and insulin resistance during diet-induced obesity. And with that, I'd like to thank everyone in our lab, especially our BLIMP1 team, Gio, Xiaoyao, Alan, Alex, and Zach, as well as our collaborators from the Department of Immunology and the Department of Medicine that made this project possible. Please come visit my poster. I have a lot of additional data there that we didn't have time to cover in this presentation. And also, if you have any additional questions, I'm always available through email at lisa.beppu@pitt.edu. Thank you so much.